Imagine yourself in the middle of an intense workout. What do you think is happening inside your body to produce energy? If you thought glycolysis, because you saw the title of the video, let me tell you, you are absolutely correct. So let's get started with what glycolysis actually is. As you can see, the term glycolysis is made up of two words. Glyco, which refers to sugar, and lysis, which means breakdown. So glycolysis is basically the breakdown of a 6-carbon sugar, which is glucose, into smaller 3-carbon molecules of pyruvate, and of course with the release of energy, in the form of ATP. The breakdown takes place in the cytoplasm of the cell, and as the process does not require oxygen, it occurs in almost all types of cells. The process of glycolysis looks quite complicated and boring. But you can remember it in a fun way, using just one simple line. Good grandma finds 4 dark green bananas, packed in plastic pouch. Each word of this line refers to a product of glycolysis, such as good for glucose, grandma for glucose 6-phosphate, and so on. This makes it easier to remember the sequence of all 10 enzymatic reactions. Moreover, the process of glycolysis is divided into 2 phases. The first phase uses energy or ATP, and the second phase generates energy. Let's begin with the first step of energy investment phase. In this step, glucose is phosphorylated by the enzyme hexokinase or glucokinase to form glucose 6-phosphate, which is a more reactive form of glucose. As you can see, in this step, ATP is used, and because one phosphate is removed, it is converted to ADP. Here's another tip for you. Whenever you see the word kinase, just remember that it is related to ATP or phosphorylation. In the second step, glucose 6-phosphate is converted to fructose 6-phosphate by the enzyme phosphohexose isomerase. Here's another thing to remember. Whenever you see enzyme isomerase, it is related to isomers, which have same molecular formula, but differ in the arrangement of the carbon chain. In the third step, an enzyme called phosphofructokinase 1 adds another phosphate group to fructose 6-phosphate, creating fructose 1,6-bisphosphate. This step also requires an input of ATP, just like the first step. This step is important, as it is the rate-limiting step of glycolysis, which means that this step controls how fast the process of glycolysis will proceed. Then in the fourth step, fructose 1,6-bisphosphate is cleaved into 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, by the enzyme aldolase. Both of these are isomers of each other. In the fifth and final step of energy investment phase, dihydroxyacetone phosphate can be converted to glyceraldehyde 3-phosphate by an enzyme, triose phosphate kinase. So, finally 2 molecules of glyceraldehyde 3-phosphate will enter the energy payoff phase. In the first step of energy payoff phase, the enzyme glyceraldehyde 3-phosphate dehydrogenase catalyzes the oxidation of glyceraldehyde 3-phosphate, which results in the formation of NADH and the production of a high-energy intermediate molecule, 1,3-bisphosphoglycerate. Here's another tip. Whenever you see enzyme dehydrogenase, just remember that hydrogen is removed and given to any electron carrier, such as NAD in our case. Also remember that, as 2 molecules of glyceraldehyde 3-phosphate entered this phase, so, each product of this phase will be considered double. In the next step, the enzyme phosphoglycerate kinase transfers a phosphate group from 1,3-bisphosphoglycerate to ADP, resulting in the formation of ATP and 3-phosphoglycerate. So, 2 ATP molecules are generated. This step is also known as substrate-level phosphorylation. Notice that, whenever the phosphate group is transferred to ADP to make ATP, the reaction is known as substrate-level phosphorylation. Then, the enzyme phosphoglycerate mutase converts 3-phosphoglycerate into its isomer, 2-phosphoglycerate. Remember that, mutase enzyme is usually responsible for the movement of functional group, as in this case, the phosphate group moved from 3rd to 2nd position. In the next step, the enzyme enolase catalyzes the removal of a water molecule from 2-phosphoglycerate, resulting in the formation of phosphoenol pyruvate. Finally, the enzyme pyruvate kinase transfers a phosphate group from phosphoenol pyruvate to ADP, resulting in the formation of ATP and pyruvate, the end product of glycolysis. This reaction is also known as substrate-level phosphorylation, as we discussed before. Overall, 2 molecules of pyruvate are produced. So, this sums up the process of glycolysis. Let's take a look into the energy yield. As discussed before, 2 ATP molecules are used in energy investment phase. Whereas, in energy payoff phase, 4 ATP and 2 NADH molecules are generated. Therefore, the overall net energy yield of glycolysis is 2 ATP and 2 NADH. Now, as you have learned the process of glycolysis, try to answer this question. Well, the correct answer is C, as phosphofructokinase 1 is influenced by several factors that determine the speed of glycolysis. I hope you understood the process of glycolysis. For more biology videos, subscribe BioMinded.